welcome to Deeksha Vedantu. I'm your Aditi Ma'am, and today we are going to do a very important chapter in this ACE series, bacha, that is cell, the building block of life. Now, if you are in 9th and when you move on to 10th, 11th, 12th, beta, if you are going to do biology, if you are going to take biology, this chapter is the building block of your biology, bacha. Okay, as it is a building block of life as well. But why do we say it's a building block of life? Beta, we know the bricks, right? We combine the bricks, it forms a wall forms a room, 4 walls, 5 walls becomes a room, 4 5 rooms become a floor, and so many floors become a building, right? In the similar manner, bacha, our body is a building, right? It looks like a building as well, right? It's a building, and it is made up of so many tissues, organ systems, and all the basics of all that. It is made up of cell, and it is called the fundamental unit of life, that it is the smallest unit with which we are made. Cells combine to form tissues to organs, and all that stuff we'll study about in this very chapter, bacha. Okay, so be ready, be very excited to learn this chapter, because this is going to give you the basics, the form of your next chapters of your whole biology as well. And if you somehow like science, somehow like biology, wants to do MBBS and all, this chapter is very, very, very important. Okay, so now let's start with cell. Bacha, first of all, understand what are human bodies. Okay, so basically humans, we are made up of cells, tissues and organs, basically. And what is the structural organization of us? We are called organisms, right? You must have heard this term in your previous science lectures or classes, right? We are called living organisms because we are living, we are breathing, we are growing, and we are organisms. And what are organisms made up of? Organisms, bacha, are made up of organ systems. Organ systems are made up of organs. Organs are made up of tissues, and tissues are made up of cells. And this cell is what we are going to study in this complete chapter in a very basic manner, in a very lovely manner that you will understand anywhere, that you will not understand anywhere, right? If you have not understand this chapter in your school, in your coaching, anywhere, bacha, this video is going to be very, very important, because I've got you so many diagrams and understandings, and we will understand it with a very, very lovely manner. Okay, now what is exactly a cell? Ma'am, cell is a brick. Okay, but brick is also made up of so many things, right? You heat it up in that, we call it bhatti. You heated that in that oven, it is made up of some site or some sort of sand. You put water in it, and it's had so many structures in it, right? So what is that brick? Is what is a cell. Now this cell, ma'am, is it the same cell we use in the physics, you know, that battery? No, that is physics cell. We today are going to study the cells of biology that we are made up of, right? Understood? Now what are these? These cells are the smallest biological unit. These cells are the smallest means nothing is more smaller than this cell. Now let me tell you one thing. Cells are also made up of so many things, and you will say, ma'am, then how is cell the smallest thing? Because bacha, those things come together and make a complete cell. They cannot exist individually. Okay, cell exists individually. So cell is the basic biological unit, and it is made up of so many cell organelles we will talk about, and combining all these cells, our living organisms are made up of. So it is a smallest biological unit, and make up all living organisms. It is a fundamental and structural unit of life. It is the fundament of us. It gives us structure, and it is the smallest biological unit. Understood? Clear? Right? It is the smallest unit. Up till now you can understand that. Okay. Now moving on, let's understand a little history of how the cell, where the cell term came from actually. So Robert Hooke in 1665 once was sitting and was watching a tree, and I was like, what is this tree made up of? So he did what? He cut the bark of the tree and the upper sheet of the tree, right? He put it under the microscope and he saw a structure like this. And he saw that the structure is made up of small blocks. What are these blocks? So he named these small blocks, as cells. And this whole upper part of that bark is called cork. So he named it as cork cells. Okay. So he observed thin slices of cork under a microscope and saw tiny box-like structures, which he then called cells. Understood? So who first discovered the cells? Robert Hooke. But then there came another scientist. He was Antonio van Leeuwenhoek. Now what did he do? Now bacha, there are two types of cells. These cells, the cork cells, they were dead. They were not growing. They were not doing any work. On the other hand, the cells inside us, they do work. They are living, right? That's why we are living. So Antonio van Leeuwenhoek discovered the living cells, the first living cell. How will you remember? The L in Leeuwenhoek is for living cells. Understood? So if they ask the first living cells, Antonio van Leeuwenhoek. If they just ask you cell, then it is Robert Hooke. Okay, understood? So he discovered the first living cell in 1674. Then now came another scientist, two more scientists came, and he discovered nucleus. Ma'am, what is nucleus? See, nucleus is called, we'll study about nucleus as well, but basically, what is the brain of our whole body? It's our brain, it's in our head. Similarly, cell also has its own brain, and that brain is called nucleus of the cell. And who discovered that is our very own Robert Brown. Okay, then came another scientist, that is Purkinje. Now Purkinje gave protoplasm. Ma'am, what is the protoplasm? See, we studied that this is a dead cell, and we studied that there is a living cell. So in this cell, in one single cell, we have some different parts. Okay, the living part, the dead part. So the living part of the cell, is called protoplasm. Bacha, understood? Protoplasm is the living part of the cell. Now a very important thing came, that is our cell theory. What did cell theory said? Cell theory said that Matthias Schleiden came and he said that all the plants are made up of cells. We are like, okay, all the plants are made up of cells. Then Theodore came, and Theodore also said that all the animals, all the living things are humans, lion, tiger, whale, all of those organisms are also made up of cells. And we are like, okay, so all the plants, all the living organisms, everything on earth is made up of cell. Understood? Then came Rudolf Virchow. What did he say? Rudolf Virchow says that every cell is made up of its previous cell, and that previous cell is made up of its previous cell. How? What are you saying? It means that we have a cell. Okay, we have this cell. Done. It is made up of which? How did it originated? It came from a previous cell which broke into different cells. Right, which broke into 2 or 3 different cells. Where does this cell came from? It also came from a previous cell which broke into 2 or 3 cells, which broke into 2 or 3 another cells. Where this cell came from? This came from another cell. Okay, so this is what happens. A cell breaks down, it makes new cells. That new cell again breaks down, makes new cells. That new cell again breaks down and makes new cell. And this whole phenomena was given by Rudolf Virchow, right? So studying about all the scientists that gave their own criteria for cell theory. Some discovered what is inside the cell, some discovered the cell itself, and some said where the cell came from. That is incorporated in this whole table. Right? So in 1665, Robert Hooke came. He first observed the cell, but that was a dead cell. Then 1674, Antonio van Leeuwenhoek came and he discovered the first living cell. Okay, then Robert Brown came in 1831. He identified the nucleus and he said that nucleus is a distinct part in the cell. Done. Then came Purkinje, and Purkinje said, and he coined the term protoplasm, the living part of the cell. Done. Then came in 1838 or 1839, both they came, Matthias Schleiden and Theodor Schwann. They came and they said that plants as well as animals, all of these are made up of cells. And then at the last, Rudolf Virchow came and he said a term, omne cellula e cellula, which means every cell arises from a pre-existing cell. Okay, that is your complete cell theory. It is very important. They can even ask you questions from it, right? Questions, and they can ask you MCQs as well. Who coined the term protoplasm? Who discovered the cell first? Who discovered the living cell first? So you should know all these. Okay, these years are not very important, but the names and their contribution, what they contributed, that is very, very important, bacha. Okay. Now, moving on next, these cells are made up, we say that these cells make up of all the organisms, but we all are different, right? I am a human being, you are a human being, there are so many animals out there, there are so many plants out there, bacteria out there, fungi out there, yeast there. What are all these made up of? Are there in all these organisms, in all these plants, are the cells same? The cell size, the cell shape, their function, their look, do they look all the same in me and a plant? They don't, obviously, right? Because if cells look the same, probably me and a plant will look the same, right? But we don't. So we classify organisms first, and then we study about the cells of those organisms. Understood? So there are, based on cell composition, how many number of cells are in an organism, we classify organisms into two parts. First is unicellular organisms. From the uni, the word uni means basically one, right? So organisms which only have one cell in them, they are made up of only and only one cell. Which are those organisms, ma'am? We are made up of so many millions of cells, but there are animals, there are small bacteria out there which are made up of just one single cell. What are those? We have amoeba, we have bacteria, yeast, paramecium. All these organisms have only one cell in them. Okay. On the other hand, we have multicellular organisms like me, like plants, like other animals. So organisms made up of more than one cell. Even if 2 cell, 3 cell, you won't call them dual cellular organisms, right? That's not a term. It's simply unicellular, that is one cell, or multicellular, that is more than one cell. Understood? That is the clarity about what organisms are and how the number of cells in the organisms classify them into different things. Now coming on to cell shape. Now beta, the cells are of different shape. Why was I drawing the cell like this, right? We will understand that, but cell is not just a round structure. It can be elongated like this. These are our muscle cells. It can be a fat cell like this, and it has a nucleus inside it. It also has a nucleus inside it, right? You just study what we are seeing, okay? We are seeing so many nucleuses in there, and we'll study them later in detail in this lecture only. Don't worry, okay? So we have small blood cells. These can be round, these can be elongated like a ring structure, right? And this is our bone cell. It has branches out there. This is our nerve cell. Nerve cell is the longest cell in our body. What is the longest cell in our body? It is our nerve cell, right? Ovum is the largest cell in our body, which can be seen in our female bodies, right? So ovum cell is the largest and longest are different, okay? Longest is the cell is long. Largest is the diameter of the cell is very large, okay? Then we have sperm cells, which can be seen in males. So all these cells have different shapes. Understood? This is round, this is a fringe like structure, this is a road like structure, and then we have this elongated cell, a fat cell. So number of cells in different organisms can be different. Cell shapes in different organisms can be also different, okay? Then moving on to, we have what is the smallest cell in the world. The smallest cell in the world, bacha, is a bacteria called Mycoplasma genitalium. It is called Mycoplasma. It is the smallest living cell. It is the smallest living cell in the world. And which is the largest living cell in the world? It is the ostrich egg. Ostrich egg is the largest living cell in this world. Understood? You'll remember this. Okay. Moving on, we have different components of cell. Now I told you that cell is made up of so many things, right? There are so many stuffs inside the cell. We have, okay, let's say this is our cell. Done. This is our cell, and in this cell, beta, we have so many things. First is, all of these cells, we have protoplasm, we have plasma membrane, we have cell walls. So we'll study all about that, but what exactly it looks like, let's see a vague idea of that. Okay, so this is your cell. This part which I have just drawn, it is called plasma membrane. It is called plasma membrane. We'll study all about it, don't worry. So basically, plasma membrane is a boundary of the cell. Now your cell is not like a random structure, right? It's not like blown like everywhere in your human body. It's a confined structure. It has a proper boundary, and inside that cell we have lots of other things that are functioning, that have their own functions. Some regulates the energy of the cell, some regulates the food, some store the food, some gives us energy, some takes energy to function, right? So all those things are collectively called cell organelles. Those are called cell organelles, and all are present inside our plasma membrane, or we call it cell membrane, and this acts like a boundary of the cell, so that not anything can enter into your cell or exit into your cell. It decides what will enter or what will exit from your cell, right? Now, if you have studied cell before or plants before, you will know that in plant cells we have something called cell wall. Animals, organisms like us, we don't have cell walls, but some of the cells, like plant cells, they have a cell wall like this outside the plasma membrane. Outside the plasma membrane, something like this, bacha, we have cell wall. We have cell wall, understood? And this cell wall is very, very rigid. Very rigid means it does not change its shape, it does not has its own brain or anything. And a very interesting thing, this cell wall is dead. Yes, this cell wall is literally a dead part of this whole cell. Understood? And then inside we studied that it has a nucleus. This nucleus should not be at the boundary. It can be at the center as well. It can be present anywhere in this whole cell. Let's just place it anywhere. Okay. So this cell, this part of inside the cell, this is called nucleus of the cell, and nucleus, bacha, is called the brain of the cell, as we saw before. Right? Now inside the cell, there are so many cell organelles. There are so many cell organelles. See, they look like this. We'll understand. We'll study all about this. Some look like this, a donut shaped cell organelle, a big structure like cell inside the cell. So all these things, such as your lysosomes, your protoplasm, all those things are inside the cell. Actually, protoplasm is a part of the plant cell. From P, protoplasm. Protoplasm is the part of plant cell. However, all this structure, see, there are so many parts in structures inside the cells, right? All those are called cell organelles. Those are collectively called everything. All these are collectively called cell organelles. And the part where these are present, so this is whole structure, it is filled with a jelly-like substance. So it is filled with a jelly-like substance, which, bacha, is known as, let me fill this color. Okay, it is known as cytoplasm. 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 Now let's study all of these one by one, starting with our first is plasma membrane, right? Starting with the plasma membrane or cell membrane. What is this? It is the outermost covering of a cell. We saw it is a boundary. It is an outermost covering of the cell. In the animal cases, in the living organisms, it is the outermost covering. However, in the plants, cell wall is the outer covering. Then it permits the entry and exit of some material. We need food. My cell needs food. So if it thinks that this food is good and I need energy from it, the plasma membrane will let that food pass into our cells, right? And let's study about this. Okay. First, okay, plasma membrane, right? The plasma membrane is the boundary layer that encloses every cell, correct? It controls what enters and what exits the cell. Very basic. Now it is composed of lipids, ma'am. These lipids and proteins help this wall, let's say, to be very strong and helps to decide what can enter and what can go out, right? So this is called plasma membrane or cell membrane. Two of the names of the same structure. It is the same structure. It can be just called as two of these names, plasma membrane, cell membrane. Okay. Now outside this plasma membrane, we have cell wall. It looks something like this. It has these various kinds of proteins on its structure, which helps to decide which can enter and what cannot enter into our cell. Understood? Now outside this cell, plasma membrane, see, outside this plasma membrane we have this structure, and that was called cell wall. Now what is this cell wall? First of all, it is dead. It is non-living, and it is the outer covering of plasma membrane, and it technically provides shape to our cell. Since it's very rigid, it helps to maintain the shape of our cell. In everywhere, in plants, cellulose is the cell wall. The name of the cell wall is cellulose. In bacteria, it is called peptoglycan. Right? In fungi, we have chitin. In animals, we don't have cell walls. Right? We studied about it, and it is dead, it is rigid, it is non-flexible and give the shape to the cell. Understood? Very basic. Then we studied about plasma membrane. Then, bacha, we have, as we said, that plasma membrane is used to take in food inside the cell and sometimes throw the stuff, the dead organelles and stuff outside of the cell as well. Or it has to give energy to our body. The cell makes the energy. It has to make the energy and put that out in our body as well. So it transfers energy, it transfers food, it transfers water, it transfers oxygen. But how do they do that? We have some transport mechanisms for that. Okay, first is called passive transport and the second is called active transport. Let's study what these transports are. Okay, now let's say you have this cell. This is your cell. Okay, and you have this some kind of substance which is concentrated more inside the cell than outside. Now what is concentration? Concentration is simply the amount of substance. Let's say this is water. So the concentration of water inside the cell is clearly more than outside the cell. Right? Now if this water moves from high concentration to low concentration, from where? From high concentration to low concentration, then it will be a passive transport. High concentration to low concentration, it will be called, bacha, passive transport. Okay, it happens very spontaneously. It happens for solids, liquids, gas, all of the stuff from higher concentration to lower, from higher to lower. So all this water that was inside the cell, it will go from higher to lower, that is from inside to outside. Understood? Active transport is just the reverse of that. Active transport in which, let's say this is a cell, again there is a water inside the cell. Now this is high concentration. Here the water is very high in concentration. Here the water is low. But still it is moving from low to high. From low to high, that is called active transport. Okay, it moves large molecules and ions from lower concentration to higher concentration. Passive transport, high to low, high to low. Lower active transport, low to high. Okay, Understood? Now, this passive transport again has so many types of it. Let's just understand first the difference between active and passive transport. They can directly ask you a question, what is the difference between passive transport and active transport? So passive transport is what? High to low concentration. Active transport is low to high concentration. Basic. Now you tell me, it is easy to go from higher to lower concentration or from lower to higher concentration? Higher to lower, right? There is so many stuff here and small chotu stuff here. What will transport better? From high to low, it will transport easily. So that means no energy is required for the transportation from high to low concentration, right? But in this place, there is already so much water and you want to add more water. You have to provide energy. You have to energize the system to move from low to high concentration. Right? Understood? So that means active transport that goes from low concentration to high concentration, it requires energy to do that. That energy is in the form of ATP, adenosine triphosphate, that is made inside the cell. We'll understand about that. Mitochondria makes ATP, right? So we'll understand about that too. Okay. Now passive transport is slow movement. It is slow. But since active transport is using energy, it is a rapid movement, right? And in passive transport, only small molecules are transported. But in active transport, large molecules can also be transported. Understood? Because we are giving energy, so we can send large molecules as well. Done? Now, moving back to the transport system, we have two types of passive transport system. First is diffusion and second is osmosis. Understood? Now, osmosis, bacha, is a very important topic in your physics as well, in your chemistry as well, and in your biology. So make sure you understand it with very focus, okay, with pure attention. Now we'll start with diffusion. We studied in passive transport the structure, the thing, it goes from high concentration to low concentration. It is the basic. That means in diffusion as well as in osmosis, our whole substance is going from higher to lower concentration. That is fixed. Okay, now studying about diffusion. So this is oxygen, this is your cell. Now your cell thinks, okay, I do need oxygen, we need oxygen to survive. So let me take this all in, right? So it takes all this in, it lets it come inside your cell. Understood? And it goes from higher concentration, here it was higher, here it was low, so it goes from high concentration to low concentration. That means diffusion of oxygen happened inside your cell. Can you say that? You can say that. Now we have food. Also your cell also needs food. So it says, okay, it's lots of food is there. Let me just take it in. So again, it travels from high concentration to low concentration. All the food goes in. So diffusion of food happens, right? Then there is one more thing. Now understand very carefully. Okay. This was food. This was oxygen. Now we have water. Again, this is high concentration, this is low concentration. So according to passive transport, it goes from higher concentration to lower concentration. So it should go outside. Okay, all the water molecules have gone outside. Ma'am, it is also diffusion. Wait. It is not diffusion now, but the structure is same. What are we missing? We are missing that this is water now. In the case of water, we say osmosis happens. So whenever you see water being transported from high to low concentration, whether from inside to outside or outside to inside, it is not a case of diffusion. I will not say that this is diffusion. I will say that it is rather osmosis and not diffusion. Why? Diffusion is a case of solid, liquid and gases. However, water is a special case of diffusion. Okay, of course water is also a liquid, but if you have some other kind of liquid, it will be called diffusion. If you have water, it will be called osmosis, right? It will be called osmosis. Now there is one more condition of osmosis that we have missed, right? During this diffusion, you can also say, okay, this is a cell, and inside the cell you have so many water on this side of the cell and small concentration of water on this side of the cell. Now what will happen? Passive transport, high concentration to low concentration. Now this is high concentration and this is low concentration. Mind you, both of these water molecules are inside the cell, right? They are not outside the cell. So this water, it can transport from high to low, high to low, high to low. Understood? Great. Now this is called diffusion. Ma'am, what are you saying? This is water. Yes, water, but see very carefully. During this transport, it is not crossing an SPM. What is an SPM? SPM is a semi-permeable membrane, right? This membrane needs to be passed. This membrane needs to be passed whenever osmosis is happening. If the water is traveling from high to low concentration but is not passing, it is not crossing an SPM, that is your semi-permeable membrane, it is not passing that is not diffusion. What is the semi-permeable membrane? Let's go back to our plasma membrane. That is a semi-permeable membrane. So every cell has plasma membrane, right? Every cell has a boundary. That plasma membrane or cell membrane is called SPM. So this water here is crossing SPM. This is our SPM. This water is crossing SPM. That is why it is called osmosis. But here it is not crossing any SPM, right? It is not crossing any SPM. That is why it is not called osmosis and it is just diffusion. This is diffusion and not osmosis, bacha. Understood? Now see here. This is our SPM. This is our SPM. Now water is going from high concentration to low concentration, high concentration to low concentration, and it is passing the SPM while going from high concentration to low concentration, and hence it is called osmosis, right? And not diffusion. So diffusion is simply the transfer of substances from high concentration to low concentration. On the other hand, the transfer of water from high concentration to low concentration, while it crosses SPM, it must cross SPM. That is called osmosis. Done. So if in paper they'll ask you what is diffusion, you can simply write it is the spontaneous movement of substances. Which were the substances? Solid, liquid and gas, from a region of high concentration to a region of low concentration, right? Substances like carbon dioxide or oxygen can move across the membrane by diffusion. Then osmosis on the other hand is defined as the spontaneous movement of solvent or water. Beta, solvent is something in which you put something, right? So in chemistry you will study this. There is a solution, and in the solution, let's say we have a sugar solution. So in sugar solution there is so much water and we add a little bit of sugar. So water is our solvent, sugar is our solute, right? So that solvent, so majorly during 9th 10th classes we'll take solvent as water only. We can take another solvents as well, but that will come in higher classes, don't worry. Okay, so we call that the spontaneous movement of solvent or water molecules from the region of high concentration to the region of low concentration through selectively permeable membrane. This is also called semi-permeable membrane. Through this SPM, it is called osmosis. Now, this osmosis, we understood what osmosis and what diffusion is, right? Now this osmosis, bacha, is of two types. Okay, it can be endoosmosis, it can be exoosmosis. Now what are these endoosmosis, what are these exoosmosis? See, in endoosmosis you will remember something is entering from endo, you remember it is entering. Okay, so this is osmosis. So definitely we are talking about water, and it is either going inside the cell or outside the cell while crossing selectively permeable membrane. Now if this water is going inside the cell, if it is going inside the cell, which one is inside? Yes. So the water will go from high concentration to low concentration because it's a passive transport, and since it is going from higher to lower while traveling through SPM, it is going inside the cell. It is entering the cell. That is why this will be called endoosmosis. This will be called endoosmosis. And if the water is going from inside of the cell to the outside, that is, it is going again from, it will go from high concentration to low concentration, bacha. Now inside there is high concentration, outside low concentration. So it is exiting the cell. The water is going from inside of the cell to the outside. It is exiting the cell, right? So it is called exoosmosis. It is called exoosmosis. If it is exiting the cell, exoosmosis. If it is entering the cell, it is called endoosmosis. Done? Very simple, just the basic concepts we have to study. Now let's see quickly the difference between diffusion and osmosis. Diffusion happens from any liquids, gases as well as solids. For osmosis, we only use water, okay, or any other solvents. Then it may or may not cross a membrane. See, in diffusion, it was here crossing a membrane. This food, this oxygen is crossing a membrane. So it was crossing the membrane, but here water is not crossing the membrane. So in diffusion, the substances can or cannot cross a membrane, but in osmosis, it is sure that they have to cross the SPM, the semi-permeable membrane or selectively permeable membrane. Then, now diffusion example is O2 entering a cell or CO2 leaving the cell, or osmosis is water entering through a SPM or water exiting through a SPM. Entering is called endoosmosis, exiting is called exoosmosis. Understood? Now related to this beta, we have to see a very important concept, that is your hypertonic, hypotonic and isotonic solutions. Now again, the word came is solution. What is a solution? Okay. Look at this. This is a solution. Okay. Now we said we have sugar solution. If you have not studied solution yet, look at this. So there is lots of water in this beaker. Okay, there is lots of water. Now this is water, and inside this water we are adding 1 spoon of sugar. We are adding 1 spoon of sugar. Now this sugar, which is less in quantity, this sugar is called solute, and the water, which is so much in quantity, it is called solvent. It is called solvent, and together this whole thing is called solution. So we typically call it water or sugar solution, salt solution. Okay, so we call this solution. So this is the basics of solution. Now let's move back to what we have to study in this particular segment is hypertonic solution. What is a hypertonic solution? Hypertonic solution, isotonic solution. This is very confusing, but I'll give you a very good trick. Okay, it is widely used, but you will remember as well. Okay, now beta, this solutions, these are comparative quantity. What do you mean by comparative quantity? Let's say I have this solution. Okay, and in this solution, let's say this is water. Okay, this is water, and I put 1 cell, I take 1 cell out and I dip it into the water. Done. Till here clear? We take 1 cell, we dip it into a water. Now see, here outside in the solution, in this whole solution, not the cell, okay, in the whole solution, solute, the solvent is very less. That means when I say comparative term, that means the solvent is very less. What does that mean? That means the water in this solution, is very less, but the water inside the cell is too much. The water inside the cell is too much, but the water inside this solution is very less. Solute is more. What I mean by that? I mean that you take a solution and you put so much sugar and just little bit of water, and in that solution you put your cell. Now inside the cell you have a decent amount of water. But in the sugar solution, where you put so much sugar and a little bit of water, the water is less. So if you compare the water in this solution and the water in the cell, that means the water in hypertonic solution, the water inside the cell is more than water inside the solution. Understood? We have a solution that is hypertonic, we put a cell in it, and if you compare the water inside the cell and the water inside this solution, you will see that the water inside the cell is a lot more as compared to the water in the solution. Now, according to passive transport, what will happen? The water will go from higher concentration to lower concentration, higher concentration to lower concentration. So, the concentration of water inside the cell is too much, right? It is high than compared to the water outside the cell. Outside the cell, water concentration is very less. So, it will go from high to low. That means it is going from inside the cell to outside the cell, inside the cell to outside the cell. And when anything or when water goes from inside the cell to outside the cell is called, it is exiting the cell, it is called exoosmosis. It is called exoosmosis. Done? Now, you tell me, if water inside the cell goes out from the cell, what will it do? It will shrink, right? There was so much water inside the cell, all the water went out, so the cell will shrink, right? The cell will shrink. That means if you put a cell inside a hypertonic solution, the cell will shrink, bacha. Done? Now moving on to hypotonic solution. It is just the reverse of hypertonic. Very easy. Now here the solvent is more. That means water inside this solution is too much. The water out in this solution is too much and water inside the cell is very little. The water inside the cell is very little, water outside the cell is too much. That means the water here has high concentration, it has low concentration. So in passive transport, in osmosis, the water will go from high to low, high to low. So high is outside, low is inside. So it will go from outside to inside, outside to inside, from higher concentration to lower concentration. Now what is happening here? The water is entering the cell. The water is entering the cell, bacha. From enter, we know it is endoosmosis. It is endoosmosis. Understood? And if a water goes inside the cell, it will swell up, right? It will swell up. So that means in hypotonic, the cell will swell up. In hypertonic, the cell will shrink. And in isotonic, nothing happens. In isotonic, the water inside the cell is of same concentration than water outside the cell. So nothing happens, concentration is same. The number of water going out is same as the number of water coming in. So there is no endo or exoosmosis in isotonic. Now, how do you remember this? How will we remember this whole thing? You will remember with this simple trick. Now see, in hypertonic, you see we use this word R, right? So from R, we remember that this shrinks. And how will I remember, ma'am, this shrinks? Because shrink also has R in it, right? Shrink also. See, this is the silly tricks we use to remember stuff. Right? So in hypertonic you have a word R, and in shrink you also have R, so you just relate them. In hypertonic, cell shrinks. That means the water is going from inside of the cell to the outside of the cell. It is exiting the cell. That means it is exoosmosis. Understood? Then for endoosmosis, the other one is endoosmosis or hypotonic. That means in hypotonic solution, what is happening? Endoosmosis, or cell is getting swelled up, right? You just remember hypertonic, shrink. The other one is the same. But if you want to remember, this is hypotonic, right? So we have O in this hypotonic, right? We have O in this hypertonic, right? So from this O, we remember it becomes big. When something swells up, it looks like O, right? It looks golomolo, it looks like O. So from that, we will remember that it will swell up. It will swell up, right? If it's swelling up, that means water is going inside the cell, water is entering the cell. That means endoosmosis is happening. Understood? Okay, this was the whole hypertonic, isotonic solution segment. Let's move on to the next one, and the very next topic we have is plasmolysis. That's a very small topic that relates to plant biology, and that is botany, right? So from plasmolysis, the P of plasmolysis, that means it is for plant cell. Okay, that means it is from plant cell. That is how you will remember. Understood? Now, what is plasmolysis? Now see, when you put a cell inside a hypertonic solution, from that R, we know it shrinks, right? Now, cell is shrinking. But if you put a plant cell inside the hypertonic solution, what will happen? Do you remember that cell wall was very rigid? It was very stiff. It was like, I won't move. Now we have put it in hypertonic solution and cell wall is still rigid and it's saying, I won't move. You go anywhere, I won't move. Okay, so cell wall is not moving. But inside the cell wall, we have plasma membrane, we have nucleus. All that will shrink. But cell wall will remain here. Look carefully. This is the whole cell. Okay, I put it in a hypertonic solution, it has started shrinking. So see, first this cell wall is directly in contact with plasma membrane. Now this plasma membrane is shrinking, cell wall is still rigid, cell wall is not moving. Then also it is shrinking more. See this green part, it is shrinking even more. Cell wall is not moving again, and then it shrinks and it forms a goal structure or a small structure, and that is this whole stuff, this whole phenomena is called plasmolysis. But this is our nucleus, plasma membrane, cell organelles, cytoplasm. It becomes small, but this cell wall stays rigid and it does not move, right? So this whole plasma membrane got detached from the cell wall, and that phenomena is called plasmolysis. Then the detachment of plasma membrane from the cell wall caused by water loss, caused by shrinking or water loss when a plant cell is placed in a hypertonic solution. Understood? So when you place a plant cell inside a hypertonic solution, it will shrink. That means that plasma membrane detaches from the cell wall because cell wall is rigid and it's not going to move. Then okay, that is all about plasmolysis. Now let's study about the parts of our cell, that is nucleus. Now nucleus, I said, is just like our brain. It is the control center of the cells, right? And bacha, have you heard about DNA? We will study about DNA in few minutes, okay? So this nucleus hosts the DNA of us. It hosts our DNA. It is the controlling center of the cell and houses the cell's DNA and coordinate activities such as growth, metabolism, division. We know cell divides. We studied about that, right? We know cell controls our metabolism. It gives us energy to do the work, right? So all that is coordinated by nucleus because it is the brain. Just like CPU is the brain of the computer, our brain is the brain of our whole body. It controls our neurons, it controls our structures, right? Everything is controlled by nucleus. Okay, now what does this nucleus looks like? First of all, it has a double membrane. What does that mean? We studied about plasma membrane, right? But sometimes this nucleus has two membranes. This nucleus has two membranes. This is a whole cell, okay? This is a nucleus. This nucleus has two membranes, and that is called a nuclear envelope. right? It has double membrane envelope, and this membrane, bacha, it has pores in it. It has pores in it, something like this. It has pores in it, and through these pores our ribosomes travel. What are ribosomes? We will study, okay? Through these pores, stuff goes in and out of the nucleus. Done. So this is the controlling center of our brain, and it houses our DNAs, controls the activities like growth, metabolism and division. And very important part, this has a double membrane which has pores in it, and this is called a nuclear envelope, right? And in this is the structure of nucleus. You can see there are so many pores on this membrane, right? And this is a nuclear envelope. This has double membrane. You can clearly see the double membrane, right? So this is a nuclear envelope, and something like this we have nucleolus. Then we have nucleoplasm. Just like we studied protoplasm, just like we studied cytoplasm, there is a jelly-like substance inside the nucleus as well, and that is called nucleoplasm. Then next, moving on to DNA. Now we said that our nucleus has DNA. What is DNA? Ribose nucleic acid. Now what is the use of this DNA? First of all, this DNA, bacha, this DNA is like this double-stranded structure. Because you've seen this in movies and stuff, right? They use it to match your DNA with your parents, and to see if the DNA of some body matches with the living person or not, that kind of stuff, right? You have seen. So this is a double-stranded polymer, and it stores our genetic information. What kind of information? The shape of your nose, the shape of your lips, the structure of your thumb, your eye color, right? Your hair texture, everything. You get this, and you'll see you have a resemblance with your parents, right? So parents transfers this DNA from themselves to you, and this is a genetic information. It holds all the information about your the way you look, the way you sound, okay? And this information is passed during sexual intercourse. Then so that is basically DNA, and it is inside the nucleus. So this is a cell, this is a nucleus, and inside that nucleus this will be something like this. This will remain in the nucleus something like this. Done? Okay. Now also we have one more structure, that is RNA. That is not basically in your syllabus, but it is RNA, that is ribose nucleic acid. In DNA we have a double-stranded polymer. In a ribose nucleic acid, that is RNA, we just have a single-stranded structure. Now based on that we have two structures, two types, that is chromatin and chromosome. Now we studied that nucleus helps in division as well, right? So what happens, beta? The chromatin is a loosely packed DNA. That means it is just inside the nucleus like this. It is inside the nucleus like this, right? It is not packed in a structure. It is not like that structure that you see in movies. It's basically inside the nucleus like this, okay? So it's a loosely packed DNA plus proteins appears as a fibrous network, and it is dispersed through the nucleus. It is all over in the nucleus, and this DNA also holds proteins as well. Done. Now during cell division, when this cell is dividing, during cell division, when this cell is dividing, this same DNA takes the form like this, takes the structure like this. Understood? The same DNA which was dispersed through the nucleus takes a structure like this or during cell division. This is a highly condensed DNA. It's a highly condensed structure as compared to this one, and proteins visible during cell division. The fibrous network, it is called chromatin, and the structured highly condensed structure, it is called chromosome. We'll study about chromosome as well, right? X chromosome, Y chromosome. That happens when what? The fetus gets the chromosome from the parents. During cell division is so much happening in the fetus. The baby in the womb, there is so much cell division happening, right? So those are chromosome comes when these chromosomes are transferred from parents to the children, that is a fetus still. Okay. Now, we have a note. In the nucleus of a single cell, there are 46 chromosomes. We have 46 chromosomes in the nucleus of a single cell. Done. Now, there are two types of cells basis on the nuclei. One is called prokaryotic cell. The another is called eukaryotic cell. Now, what are prokaryotic cell and eukaryotic cell? Let's understand from this difference, then we'll move back to it. Okay. Now, see, this is very small, 1 to 10 micrometers. Micro is beta, 10 raised to power minus 6. Okay, that is micro. Yes, micrometer is 10 raised to power minus 6. Okay, and that is 10 to 100 micrometer. Done. Then nucleus, there is no nucleus in the prokaryotic cells. I told you, we'll come back to it. Now, see, we have this nucleus. We have, what did I do? We have this nucleus. Okay, now inside this nucleus, inside this cell, see, in the nucleus is absent, DNA is in the nuclear region. See, this is a cell. Okay, this is a cell. Now we studied just before that DNA is inside the nucleus, but in prokaryotic cell there is no nucleus. Plants don't have brains, right? So this, where is the DNA, ma'am? DNA is there. DNA is everywhere. But if nucleus is not there, where is DNA? DNA is beta anywhere in the plant. It is remaining anywhere in the plant. Like this, it is just laying around inside the plant, chilling. Okay, inside the plant cell, so it is chilling. And this region, that region where the DNA is present, this is called nucleoid region. This is called nucleoid region, a region where DNA is present without the nucleus. Right? And in eukaryotic cell, what is this? It's a simple cell. In eukaryotic cell, there will be a nucleus. There will be a nucleus, and inside the nucleus, we will have our DNA. We will have our DNA. Done? Now, this prokaryotic cell, it usually has only one chromosome. It does not have a nucleus. The DNA is in the nucleoid region and it is only has one chromosome. Then on the other hand, in the eukaryotic cell that we have in us, it has 46 linear chromosomes. We as humans have 46 human chromosomes. Right? Now, in mostly it is absent, only ribosomes are present. What are cell organelles? Cell organelles, beta, we'll study them in just a minute. But in this prokaryotic cell, there are no cell organelles. Cell organelles like mitochondria, lysosomes, ribosomes, and all those. Ribosomes is not cell organelle, but Golgi apparatus and all those things are present in the cell, which are absent in this. So there are no cell organelles, only ribosome is present. Okay. Now, on the other hand, in eukaryotic cell, we have mitochondria, we have ER, that is, we'll study in a bit, don't worry. Golgi apparatus, lysosomes, chloroplasts in plants, we have all that cell organelles. Okay. Then in this prokaryotic cell, we have cell walls, that is your peptidoglycan we studied in the starting. In bacteria, the cell wall is there, right? In plants, the cell wall is there. In fungi, the cell wall is there. So, in prokaryotic cell wall is present. However, in eukaryotic cell, in plants, cellulose is present. In fungi, chitin is present, but is absent in animal cells. It is absent in animal cells. Then, and next, the very last, it has motility structures. Now, beta, this prokaryotic cells, it has these hair-like structures. It has these hair-like structures to move around and to dance around with other cells. Okay, so it is basically for the motility. For the motility is related to your motion. Okay, mobility, and that is the hair-like structure called flagella. right? It has hair-like structures called flagella. On the other hand, in eukaryotic, the motility structures are cilia or flagella. Okay, these are some of the basic things that you need to remember. No need to go into deep into that, right? Now, in the genetic material of prokaryotic cell is cellular DNA and offer plasmids. And on the other hand, in eukaryotic, we have linear DNA with histone proteins, just linear DNA. And in prokaryotic, we have circular DNA. Done? Okay, moving on, next we have cell organelles. Now we are going to study lots of cell organelles. Now, these cell organelles are organized structure or little components inside the cell. I told you, right? And all these have unique shapes, unique functions, unique structures. So inside this cell, you have nucleus, and with that you have lots of cell organelles. This, you have Golgi apparatus, you have vascula, you have lysosomes, and we'll study all about all these structures. Okay, now on the nucleus, bacha, see, this is your cell, this is your nucleus. Now on the structure, on the boundary of nucleus, you have some cell organelles like these, and these are called endoplasmic reticulum. Endoplasmic reticulum are what? These are simple membranes. See, you can see these are round in structure membranes and a network. This is a membrane bound network that lies close to the nucleus. It lies close to the nucleus and serves as the site of synthesis and processing of proteins and lipids. We need protein in our body. We ourselves also need protein in our body. And what makes those protein? These endoplasmic reticulum makes those proteins. It is a site for the synthesis and processing of proteins and lipids. And on these beta, you will see now small structures. These small structures, small pores or structures, can you say? Now what are these? These are ribosomes. So we have two types of endoplasmic reticulum. First is smooth endoplasmic reticulum, second is rough endoplasmic reticulum. Now rough endoplasmic reticulum has ribosomes on it. See, rough endoplasmic reticulum has ribosomes attached which helps in the synthesis of protein, right? These does not have. These synthesize proteins, right? Ribosomes synthesize proteins. On the other hand, this synthesize lipids. Now what are lipids? We are saying lipids a lot of the time, right? Lipids are basically your fats and hormones, right? We all have hormones in our body. Males have testosterone, females have estrogen, we have dopamine, we have so many adrenaline, we have so many hormones. Where are those hormones produced? They are produced on a site on endoplasmic reticulum near the nucleus, on the smooth endoplasmic reticulum, not on the rough. Rough one are ribosomes. They produce protein. Then okay, now we have what is the structure, what are the additional roles? They are protein folding and quality control. They helps that whatever the things, see, just like some bad humans do drugs, and just like that, if something bad enters into the cell, now it is going through the nucleus to the nucleus, right? We will attack the nucleus. Then ribosomes are like, hey, where are you going? Hey, stop right there. We will quality control you. We will check if you are a good person or a bad person, and then only we will let you enter into the life of nucleus. I wish we would have someone like that. We have our parents, we have our friends, but we don't listen to them, don't we? Including me. Okay. Then smooth ER, it helps in detoxification of drugs. Now, sometimes these drugs, we know, right, that alcohol and drugs directly impacts our liver. Our liver helps. It tries to protect us. But if you overdose it, just like some bad people do, the cell will get damaged, right? But from the point of view of the liver, of the cells, of the nucleus, they are trying to protect, and the nucleus has given this work to smooth endoplasmic reticulum, that is SER, to detoxify the drug. This helps in quality control. This helps in detoxification. Okay, now this RER, it contributes to plasma membrane protein supply, or plasma membrane actually decides what gets in the cell and what goes outside the cell. So it needs protein, it needs energy, and that is why it contributes, it needs protein, and that protein is given by these ribosomes on the rough endoplasmic reticulum. Done. And these are same, it needs lipids also, our plasma membrane also needs lipids, so they synthesize lipids for plasma membrane as well. Done. So we are done with endoplasmic reticulum, we are done with nucleus, DNA, we are done with plasma membrane, cell wall. What else is left? First is Golgi apparatus. Golgi apparatus. What is Golgi apparatus? It is like a packing and a shipping center. It is written, right? The packing and the shipping center. So whatever comes, whatever goes, it packs it and it ships it outside the cell. Right? It is a stack of flattened sac-like structures. You know sacs, right? Those bori we call in Hindi. So these are the structures. So those sacs are used to store, their sacs are used to go and like ship the stuff, right? That is what this Golgi apparatus does. Golgi, I like to call it Golgi apparatus. So these are stacks of flattened or stack-like structures. They are stacked on each other like this, right? Now what is this? Their use? Same. It modifies, it sorts, it packs the proteins or lipids into vesicles for transport, secretion, or lysosome formation. Say, correct? This Golgi is a sac-like structure which stores, modifies, and then transports the lipids, the proteins, the lysosomes, and all in our cell that is needed in our cell. So it is a packaging and a shipping center. Now, ma'am, what is a lysosome? Lysosome, bacha, these are also called the suicidal bags. You will remember these. These are called suicidal bags or digestive bags, right? These are tiny structured, tiny sac-like structures, and these helps to digest the parts of the cell which are no longer in use. Okay, this is a cell. These are lysosomes. These are lysosomes. This is nucleus, endoplasmic reticulum, and these are Golgi apparatus. Okay, so all these structures, let's say this Golgi apparatus has gone bad. It is just sitting there doing no work. So lysosome will do, hey, you are bad. Let me just send you some digestive acid, some bad things toward you so that you die. Okay, so this digests the dead or damaged cells and organic substances, some things which entered into a cell which are bad or which are dead. It digests them, and sometimes when the whole cell is dead, it bursts out. Lysosome, it bursts and ends the cell. It bursts and the cell suicides. That is why it is called suicide bag of the cell. Whenever the cell is dead, it bursts and it releases those toxic acids and enzymes, and hence the cell dies. Okay. Okay. Moving on to vacuole. Ma'am, what is this vacuole? Now, inside the cell, you'll see vacuole takes lots of space. Okay, these are large. These is a fuel-filled compartments. There is a fluid that is filled in this vacuole, and it stores. See, Golgi apparatus is a shipping center. Lysosomes is helping to kill the dead items. Now, when the Golgi apparatus is shipping those items, it has to store the items somewhere, like some storehouse. So this is the storehouse of our cell. It store nutrients, waste, and help maintain cell's rigidity. This is a cell beta, right? And this is a nucleus, endoplasmic reticulum. This vacuole takes lots of shape of the cell, and it gives its structure. It takes the massive amount of shape of the cell. Done. And it helps to maintain its rigidity. Now, some of the points is, in plant cell, a central vacuole may occupy 90% of the space of the cell, right? Pushing the nucleus to its periphery. Periphery means the boundary. Okay, and in animal cells, vacuoles are smaller and more numerous. In plant cells, normally one vacuole is there, but in animal cells, the vacuoles are smaller in size, but there can be a number of vacuoles. Okay, done? Then mitochondria. See, this is something everyone remembers if they go out to do computer engineering, if they go to do bio or not. Mitochondria is something every person on this planet remembers. Why? Because it is the powerhouse of the cell, and if it is the powerhouse of the cell, it is powerhouse of the tissue, the organ system, and hence the organism as well, right? So it is powerhouse of the cell. Mitochondria are double membrane organelles. So this is the first membrane, and then the inner membrane is this, again this this hairpin like structure inside the mitochondria. They generate ATP through cellular respiration. Now we studied about respiration. Also cells also respirate. So while respirating, we make energy for our body. What makes energy? Mitochondria in the form of ATP, adenosine triphosphate. This is very important. ATP is adenosine triphosphate. Mitochondria is the powerhouse of the cell which makes energy in the form of ATP during cellular respiration. And it is a double membrane organelle. Then what are some of the points? Outer membrane, it is smooth, it is permeable to small molecules. That means small molecules can enter into it. Inner membrane, on the other hand, see, it is highly folded, right? It is highly folded structure into cristae, that is increases its surface area. Done? The outer membrane is smooth, the inner membrane has so many folding like structures. Right? Each mitochondria contain its own circular DNA, its RNA, its ribosomes. That means it contains its own genetic information, it produces its own protein, its own lipids, and it doesn't have to look towards nucleus like, I want protein, give me my protein, and nucleus is like, I don't like you. You give so much power and you take away my function. And then mitochondria is like, okay, I'll make my own protein. I don't want your protein. Keep your protein. Okay. So that is what a mitochondria is. Allow limited self-protein synthesis. Done. Now, next cell organelle is plastids. Now, this plastid is a colorful plant organelle. Anything that has color, leaf, a flower, a plant, anything that has color, that color is because of this plastid. This is a color plant organelle. The plastids are plant-specific organelles from plant. Okay, so plastids are plant-specific organelles that house the pigments, the store reserves, and perform photosynthesis. The most important part of our plants is photosynthesis, is making food for the plants. And what helps in doing that? Plastids helps in doing that. Okay, so plastids we found in plants. This store our reserves, it helps forming the pigmentation that gives the colors, and it performs photosynthesis, right? It also harbors its own DNA, RNA, ribosomes. Now these are of three types, bacha. First is your chloroplast. We know chloroplast is found in the plants. It is a colored part of the plant, colored part of the flowers, right? Now chromoplast. Chromoplast is the it contains chlorophyll pigment, right? Photosynthesis takes place due to chloroplast. Done. This chromoplast, it contains the chlorophyll pigment. What am I doing? There are so many tongue twisters in it. Chloroplast is the colored part of the flowers. Chromoplast holds the chloroplast pigment, right? That helps to give the color. Then, now what is this leucoplast? It is a colorless plastid. It is a colorless plastid. Then ma'am, what is its function? Why it is a plastid? If it is colorless, plastid means color. But it actually stores and synthesizes our food. When after photosynthesis food is made, it does not go directly into the plant. It stores and it gives it to the plants as required when required. Okay? Now, next we have chloroplast. Let's study quickly about chloroplast. Now see, chloroplast is something like this. It is double membrane, right? It is a green color plastid. It helps in photosynthesis and it helps in synthesis of food, that is photosynthesis, right? Then imparts green colors to various parts of plants, and bacha, it also maintains oxygen concentration in atmosphere, right? It absorbs carbon dioxide, gives off O2, which is the main part, chloroplast. It helps in photosynthesis, which gives us our oxygen that helps us to breathe. Okay, it also has its own RNA, DNA and ribosomes, right? These are coin-like structures. These coin-like structures are also present in this chloroplast. Done? Okay, now it has its own DNA, it has its own RNA, it has two membranes, outer membrane, inner membrane. Done? Chloroplast is the plastid, helps in photosynthesis, it helps in synthesis of food, imparts green color to the plants and maintains oxygen level in the atmosphere. Done. Now in cell division, bacha, we have two types of cell division, as we studied in the different shapes of cell, the classifications of cell on the basis of shapes, the class, the concentration, right? But now cell also divides. We studied about that, right? Now why cell divides? Because as the cell is like a parent cell, right? So the cell divides into its daughter cells to produce more cells. That's it. Because when a cell dies, you have to have a new cell to replace that, right? That's why cell divides, right? We as human beings, we are dividing just like that, cells are dividing. Now there are two types of cell division. First is mitosis, this is meiosis. Now on the basic level, bacha, in mitosis a single cell divides into two cells. A single cell, bacha, it divides into two cells. On the other hand, in meiosis, the single cell divides into four cells. It divides into four cells. Now, very important part, that is not the only relationship or the only difference between mitosis and meiosis. This is called equal or equinational division. Okay, now why is that? Because beta, we saw that a normal cell has 46 chromosomes, right? Now during division, what happens to these chromosomes? Do they decide that I want to be with this chromosome, that chromosome? No, all the cell needs to have 46 chromosomes. So this cell also gets 46 chromosomes, this cell also gets 46 chromosomes. Now, ma'am, meiosis also, this gets 46, 46, 46, 46. No. In this is called reductional. That means the chromosomes reduces, right? So this gets half of the chromosomes. That means 46 by 2, 23. So this gets 23, this gets 23, this gets 23, this also gets 23. That is cell division, bacha. Right? Now let's quickly revise what we did. All the organelles, all the cell organelles. First was endoplasmic reticulum. It is a network in the cytoplasm near the nucleus, right? And it helps in transporting proteins and fats. It helps in synthesizing proteins and fats for the plasma membrane, right? Then we have Golgi apparatus. Golgi apparatus modifies, it packs, it's a shipping center, right? It modifies, it packs and it transports proteins and lipids that we need the body to function, right? Then we had lysosomes. Lysosome is a cleanup organelle. See beta, in our body we have so many organs like lungs, like kidneys, heart, what else we have, reproductive system, respiration system, right? All of these have different functions, just like organelles have different functions. Now if something goes bad, we go to doctor, we have surgeries, we fix them, and sometimes if someone's kidney is not working, they take the first kidney out or they replace it with some donor, right? Just like that, lysosome is the doctor, let's say, or the suicide bag as well. Now if my Golgi apparatus stops working, so what it does, it sends harmful digestive enzymes to Golgi apparatus, which helps it die. Okay, so that is what a lysosome do. It is a cleanup organelle that breaks down waste and damaged cell parts. Right? Then we have vacuole. Vacuole stores water. It's a big organelle. In plant, it's just only one, occupies 90% of the space. In animals, it is small, but too many. Right? What is its work? It stores water, minerals, sugar, waste material, then that helps to transport it. Right? Then we have mitochondria. Mitochondria is the powerhouse of the cell, right? It's the powerhouse of the cell, and it releases energy from food. The food we take, it makes energy and it gives this to our cell, to our body. Then we have plastids. Now plastids are plant organelles, right? And it helps to synthesize and store food, right? Then we studied about mitosis and meiosis. Mitosis is a cell division that forms two identical daughter cells. Mitosis was two identical daughter cells, right, from one parent cell. Meiosis that forms four daughter cells and half the number of chromosomes. Here chromosomes were same, here the chromosomes were different. And that is our one-line definitions that you can use. Now, beta, the new topics you have, the cell theory we understood, right? But what if they ask cell theory? Those are the important points. Those are basically all the living organisms are made up of one or more cells. The cell is the basic unit of basic and structural and fundamental unit of life, right, of human living beings. Now all cells arise from pre-existing cells. We studied all our living organisms are made up of cells and all the cells arise from pre-existing cells, right? This unifies all biology from bacteria to humans and explain the continuity through cell division. The life from dinosaurs to now is sustained on cell division. That's how important cell theory is, and it has been proved true again and again and again, right? Now, what new topic we had was the cell growth, right? How do cells monitor their growth to maintain a balance? See, let's read it very carefully, okay, line by line. Cells also have natural ways of dying. Now we know there are lysosomes. With the help of lysosomes, cells die. But if the cells keep on dying, we won't be able to sustain, right? We need cells to sustain. So they also grow together. The cell divides, lysosomes ends the dead cells. Cells divides, lysosome ends the dead cells. Now this balance has to be maintained. If there are so many dead cells, it results into cancer. If there are so many new cells, dead cells are still there, it also is very bad for our body, right? Now this balance is maintained by programmed cell death. Whenever a cell is not correct, it is not functioning, it is not a functioning member of our society, that is our body, it dies, and this is a genetically regulated and organized process, selective cell destruction. The cells which are bad, they are destroyed by selecting them particularly, that we have to destroy the dead cells only, we don't have to destroy the perfectly working cells. That is called selective cell destruction, right? It is essential for normal development, cellular quality control and immune function, right? When an embryo develops, the PCD helps form fingers. Okay, this is a very good part. When you say frogs have hands like this, but humans have hands like this, this is a very bad human hand, but still. So what they say is that these cells, the cells these were here, we selectively, our body, when we were an embryo, when we were this small inside our mother's womb, it selectively destroyed these cells, so we don't have a web like fingers, right? Otherwise we would have fingers like frogs, right? So when an embryo develops, the PCD helps form fingers by eliminating cells between these fingers or digits. We call it, without it we would have webbed hands, right? That is really curiously created to make you answer or question so many things, right? That is it, bacha. This is the same thing. I repeat it back. Okay, that is it, bacha. That was your whole cell chapter. I hope you understand it better. We will do so many questions of this chapter as well, so make sure you subscribe this channel and like the video. If you like the video as well, you like the structure I'm using to teach you, and that is it. All the questions will come from this video only. We will do lots of questions as well, and I gave you one AI tool as well. Go watch the video and maybe put the link of this video and generate your own notes, so you don't have to sit and write so much stuff. You have to revise by writing, but making notes will be helpful if you use that AI, right? Okay, that's it, bacha. Subscribe this channel for so many more videos. I'll meet you in the next one. Tata, bye bye.